Everything you guys say about me, like whispering into your death,
Everyone will hear.
I rely on whoever's sitting there. Like, I cannot get here early.
Actually, I already checked compliments for this round. Like, you can actually get to a minute, and there's this The color of the projector is so different from the actual PowerPoint. No. So look, I can't even reach after that up there.
Okay, so hopefully everybody remembers we have an exam on Thursday, right? That exam will cover everything since the last exam up to as far as we get today in chapter 9, which we'll hopefully be able to finish, but we'll find out. So I'm sure you guys have all heard of DNA. Everybody's heard of DNA. Everybody knows basically what it is. And you guys probably know more or less the basic structure. Everybody's heard about the double helix, kind of looks like a twisted ladder. Today we're going to go into a bit more detail than that. So, if I were to ask you this question, you guys can probably all answer it, right? Yeah, it's not a trick question. DNA. Of course, everybody knows that. But we didn't always know that. In fact, this is a relatively recent discovery. So up until about the middle of last century, we still really weren't sure. So to put it in perspective, when my parents were born, we still didn't know what the molecule was responsible for passing hereditary information from one generation to the next. So right around the middle of last century, we still didn't know the answer to this question. We did know that genes were located on chromosomes. And we did know that chromosomes were made of both protein and DNA. So we knew that the information that was passed from one generation to the next was stored in chromosomes. We just didn't know whether it was the DNA or whether it was the proteins in those chromosomes that contained the actual hereditary genetic material. In fact, up until the 40s, the case for proteins seemed a lot stronger. Can anyone guess why? Yes?
Because there were like 20 parts of the protein and only 4 parts for DNA.
Exactly. There are 20 different amino acids, and there's only 4 different nucleotides in DNA. So if you need to store a huge number of information, having a larger variety of building blocks seems to make sense, right? Of course. So if you have 20 different amino acids, you can easily make a lot more combinations than you can with only 4 different nucleotides. Does that make sense, everybody? So it wasn't until 1952 that we actually figured out the answer to this question. And when I say we, I mean those guys. So they did a really cool set of experiments involving a specific type of virus called a bacteriophage. Now, a bacteriophage is a virus that attacks bacteria. Specifically, this one was a T2, which infects the bacterium E. coli. You guys ever wondered what the E in E. coli stands for? There you go. Now, the bacteriophages are really interesting. They look kind of like a lunar lander. That's actually what they look like. And you guys should hopefully remember from earlier in the semester when I said that viruses cannot reproduce on their own. This is why they're generally not considered to be alive. Why can't they reproduce on their own? What don't they have?
Sex cells.
No. I mean, they don't have those, but you don't necessarily need those. Bacteria also don't have sex cells. Viruses don't have ribosomes, the organelle that builds proteins. Without the ability to build proteins, you can't make really anything. So the only way that viruses can reproduce is by infecting the host cell, injecting their DNA into it, and then hijacking that host cell's ribosomes to make more bacteria. As it turns out, that specific mechanism of reproduction is what allowed this experiment to be successful in the first place. It's kind of a weird way to reproduce. Injecting genetic material into somebody else, like the face huggers in the movie Alien. Have you seen Alien, or is that too old? Anyway, bacteriophage, these viruses are extremely simple. They're made up of basically only two things. They have a protein coat, and that protein coat contains DNA. That's basically all there is. This made them an extremely great model to use for this experiment. Because either the proteins or the DNA could be tagged with radioactive markers. You guys remember what those are? Radioisotopes? Yeah, of course you do. So you can tag a protein with a radioisotope that won't tag DNA, and you can tag DNA with a radioisotope that won't tag protein based on which specific isotope you're using. So this allows you to tag one but not the other, which is going to make this experiment a lot simpler. So what they did was they had two different batches of these bacteriophages, and they allowed them to infect separate batches of bacteria. One batch had been grown in a solution of radioactive sulfur to tag the protein, because a lot of amino acids rely heavily on disulfide bonds. The other had been tagged with radioactive phosphorus to mark the DNA, because phosphorus is an important component in DNA. Shortly after the infection, they blended these things up, which shook loose any parts of the viruses that remained outside bacterial cells. You guys see where this is going? So only what was actually injected by the virus into the bacteria would remain in the solution after this. Then they can spin this down in a centrifuge. The viruses being lighter will remain suspended in the liquid. Centrifuges are really cool for that. And at this point, the viruses and the bacterial cells were totally separated from each other. Then all they had to do was measure the radioactivity of the pellet of cells at the bottom containing the bacteria. And they found that when the bacteria had been infected with viruses containing the tagged proteins, the radioactivity remained mainly in the liquid and not in the bacterial cells. That suggests that the proteins from the viruses did not enter the bacteria. But when they used the tagged DNA, they found that most of the radioactivity was in the pellet of cells at the bottom instead of in the liquid. Furthermore, when these bacteria were returned to a liquid growth medium, they soon ruptured, because that's what happens after they produce enough bacteria or viruses, they just explode. And the viruses released contained a radioactive radioactive DNA, but no radioactive protein. So this pretty conclusively suggests that it was the DNA that the viruses had transferred into the bacteria, not the protein. Therefore, the hereditary genetic material must be contained in the DNA. So this is just a summary of the replication cycle of bacteria, in case anyone's more, or viruses, in case anyone's curious about how they work. There's your one that doesn't work, that's great. So basically, the virus will come in looking like a lunar lander and attach itself to the surface of a bacterial cell. It then injects its DNA into the cell. That then causes the bacterial cell to produce new virus DNA and new virus proteins, making complete new viruses, and eventually explodes and releases all the newly produced viruses. So that's how viruses reproduce. Anybody have any questions so far? I'm not going to expect you guys to know this experiment in minute detail, but you should have a very basic understanding. So by the time these guys made this discovery, we already knew a lot about DNA. We've been studying it for years. All of its atoms have been identified. We knew how they were covalently bonded to each other. You guys remember what a covalent bond is? How do you form a covalent bond? A lot of people miss that in the test. Yeah, two atoms will share an electron. What we didn't know, though, was the specific arrangement that gave DNA its unique properties. So we weren't sure how it had the ability to store information. And importantly, we weren't sure how it had the ability to easily copy information. Because DNA, to serve its purpose, has to be pretty easy to make copies of. We also weren't sure how it was able to pass information from one generation to the next. So a lot of questions were still unanswered. There was actually a really heated race to make these discoveries. It was probably a really exciting time to be in biology. Although this is too. Anyway, so DNA and RNA. These are both polymers of nucleotides. Throwing some probably unfamiliar words at you guys. So what's a polymer? There's a clue in the name. It starts with poly. What does that mean? Multiple or many. So a polymer is basically a long chain of a lot of subunits. Each individual subunit is called a monomer. So you link dozens or hundreds or thousands of monomers together to form a polymer. But then what's a nucleotide? Yeah, so a nucleotide is a molecule that has three things. It has a sugar, it has a phosphate group, and it has one of the nitrogenous bases. So this image here shows you the basic structure of a nucleotide. Here's our sugar, here's our phosphate group, and then there's one of the possible nitrogenous bases. In this case, it's thymine. There are several other possibilities. Anybody know what the other nitrogenous bases are? Who's got it memorized already? Adenine, guanine, cytosine, thymine, and uracil, if we're talking about RNA. We'll get into that in a moment. That's one of the key differences between RNA and DNA. So if you look more closely at a single DNA strand, which we can call a polynucleotide, a polymer of nucleotides can be more specifically called a polynucleotide. You see that the individual monomers are joined in a very specific way. So this is a diagram of one strand of DNA. We split the double strand in half. The sugar of one group will bond to the phosphate group of the other, and vice versa. So in the same strand, the nitrogenous bases do not chemically interact with each other. It's only the sugars and the phosphates. They bond together to form what we call a sugar-phosphate backbone, because that forms the backbone of the DNA strand, and it's just sugar-phosphate all the way up and down. Now in this diagram, we can also see the sequence of nucleotides. So pay specific attention to the fact that it's just sugar, phosphate, interacting along this chain, and the nitrogenous bases are not bonding with each other at all. What do the nitrogenous bases in the nucleotide end up bonding to? Anybody? Yeah, to the nitrogenous bases on the other strand, which is actually what holds the double strand together.
So that would be the link between.
Yeah, exactly.
So the link between is the genetic or the strand itself?
It's the nitrogenous bases. So the whole thing is the genetic material, technically. But it's the individual bonds between the individual nitrogenous bases, like the adenine and the guanine.
And there's 40.
Yeah, there's 4 in DNA and there's 4 in RNA. 3 of them are the same. So, looking again a little bit more closely at an individual nucleotide, we can see the chemical structure of the 3 components. This is a bit more detailed than we were looking at before. So you guys should recognize this thing. That look familiar from anything we've looked at this semester?
Like,
Yeah, this is the phosphate group. Where have we seen those?
ATP.
ATP. It's the exact same thing you have 3 of in ATP. So this has a phosphorus atom at its center and is the source of the word acid in the term nucleic acid. The sugar here has 5 carbon atoms. You have 4 in its ring, and then 1 extending above the ring right here. The ring also has an oxygen atom in there. So remember that this corner is oxygen, carbon, 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 and carbon there. In this case, the sugar is called deoxyribose because it's ribose. Wait, no, this one's ribose. This is from RNA. Never mind. So in this case, this happens to be a nucleotide from RNA, and we can tell because this sugar is ribose. That's what the R in RNA stands for, ribonucleic acid, from the word ribose. If it were DNA, then this oxygen atom right here would be missing, and it would be called deoxyribose because that ribose has been deoxygenated, and that's what the D in DNA stands for, deoxyribonucleic acid. It's not dioxyribose, as most people tend to think. Did I confuse everybody there, or did that help? At least now you know what DNA stands for, you can trust your friends. Any questions before I move on? Yes?
What's a polymerase?
So polymerase is an enzyme. Doesn't break down, builds them. So I mentioned earlier this semester about how enzymes are conveniently named to tell you pretty much exactly what they do. So there's several different polymerases involved in the synthesis of DNA. They're all called DNA polymerase, and then they have a number. And what DNA polymerase is used for is making polymers of DNA. Anything else? Sorry.
Is there any confusion because the A is always for life?
Nope. No, A's is for enzymes. And enzymes can break things or they can build things. So enzymes Maybe I should take a moment for that, because there were a lot of erroneous answers on the exam when I asked people what enzymes do. A lot of you seem to think that they're only involved in breaking things apart. They're also definitely involved in building things too.
They facilitate a chemical reaction.
Yeah, so they facilitate a chemical reaction by lowering the activation energy required for that reaction to proceed. That's what enzymes do. The reaction could be catabolic, or it could be anabolic. So it could be breaking something down, or it could be building something.
I was just curious, the phosphate group, so is the phosphate group acidic? Is that why?
Yes,
Okay.
yes, technically an acid. All right, anything else before we move on? Great. So a bit more about these nitrogenous bases. If you see these four, are we looking at DNA or RNA? Who read the chapter? We're looking at DNA because we have thymine. So the nucleotides are distinguished only by the nitrogenous base they contain. Other than that, they're all the same. So they all have the sugar, they all have the phosphate. The only variable part is this nitrogenous base. Now each nitrogenous base here has either a single ring or double ring structure with a few functional groups attached. Functional groups are just small arrangements of atoms on the outskirts of a molecule that affect how they function in chemical reactions. If it has a single ring structure, like cytosine and thymine, it is a pyrimidine. If it has a double ring structure, like adenine or guanine, it is a purine. Now, an important thing here, which I'll be harping on throughout this lecture, is that purines only bond with pyrimidines and vice versa. You never see an A bonded to a G or a C bonded to a T. That is what allows DNA to have a uniform width, that double helix. If you had double ring structures bonding to double ring structures and single ring structures bonding to single ring structures, the width would be variable, but it's not. We actually didn't talk about that, so forget that. That was a test. But now you know what a functional group is in general. You guys still need this? Alright. So when it comes to DNA, the main role of those functional groups, those little side chains attached to our nitrogenous bases, is to determine which other bases they can pair with. Now the nitrogenous bases pair with hydrogen bonds. So it turns out these huge double helix polynucleotides of DNA, each side is held to the other side only by hydrogen bonds, which are relatively weak, right? That's important because in order for the DNA to be copied, as we'll find later, those double strands have to be separated from each other. So if we look at the amine group attached to cytosine, that ends up bonding with the carboxyl group attached to guanine, but it will not bond with the amine group attached to adenine. So if the amine groups, those are the NH2, they readily form bonds with carboxyl groups. So the complementary attachment of functional groups makes these nucleotides or nitrogenous bases able to interact with each other. So those functional groups are responsible for one of the most important properties of DNA, and that is called complementary base pairing. This is very important, and I'll go into it in detail later in this lecture. But keep that term in mind, complementary base pairing.
So the functional groups is the functional group.
So the functional groups are the amine groups or the carboxyl groups. Like if you look up in the top row there, the carbon double bonded to an oxygen or the nitrogen double bonded to two hydrogens. That's not going to be extremely important for the test. I'm not going to ask you guys to tell me which functional groups are on which bases. Just need to know that they exist. So as I mentioned, the four nucleotides will differ only in the nitrogenous base they contain. All right, this is the one I thought I had on screen earlier. That's thymine in DNA. So everything else is identical. All the nucleotides have a phosphate group, and they all have a sugar. If we're talking about DNA, that sugar is deoxyribose. What if it's RNA? What's the sugar? Yeah, it's regular old ribose. And they're broken down into two main types that I mentioned earlier briefly. So thymine and cytosine, those are the pyrimidines with their single ring structure. Adenine and guanine with their double ring structure are called pyrimidines. And I'm going to repeat this because it's important. Purines will only bond with pyrimidines, and pyrimidines will only bond with purines. You'll never see a purine bonded to another purine. Doesn't work out. Yes. So that way you always have a total of 3 rings in the middle of your double rings, which is what allows the length to be uniform, or the width to be uniform. Everybody's comfortable on the difference between purines and pyrimidines, right? Everybody knows what they are? Okay. So as the name implies, obviously that sugar in ribonucleic acid is plain old ribose. What's the difference again? Yes, DNA has deoxyribose, which is called deoxy because it's missing this oxygen atom. That's it. Instead of a hydroxyl group, it just has a hydrogen atom. There's the difference. Also, if you see the nitrogenous base uracil, then you know you're looking at RNA. DNA has adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. Uracil is chemically very similar to thymine. So other than those two differences, a single nucleotide in RNA is identical to a single nucleotide in DNA. Just that oxygen atom on the sugar and the presence of thymine versus uracil. Those are the only two differences in a single nucleotide monomer. Oops, didn't get that button. Where do we go? All right. Now, what's another important difference between DNA and RNA? Yeah, that's another important difference. RNA can leave the nucleus. How about structurally? If we're looking at the whole molecule, yeah, RNA is a single-stranded molecule. So it doesn't exist in a double helix. Instead, it exists in a single helix. That's the other key difference between RNA and DNA. Okay, so once Hershey and Chase made that cool discovery that it's actually DNA that's responsible for the passage of hereditary information from one generation to the next, then the race was on to figure out what this super important molecule really looks like. The first to figure it out were these two nerds. A guy from America named James Watson, who was a grad student at the University of Cambridge, I think, working in the lab of the British guy Francis Crick. 1953 they made this discovery. Watson had just finished his PhD and was 23 years old. At the time when he made a discovery that got him a Nobel Prize. Crick was older, he was a senior researcher. Can you imagine that? Who in here is like 23? A couple of you? Yeah, can you imagine having a Nobel Prize already? Some people, man. Anyway, so there were a lot of people working on this discovery, and no one really quite had it yet. A lot of people were studying it, and people were taking different approaches. Now, a big breakthrough came when Watson visited the lab of a guy named Maurice Wilkins at King's College in London. Maurice Wilkins had someone in his lab named Rosalind Franklin, who was an early pioneer in X-ray crystallography, which is a really cool method used for studying the structures of individual molecules. So she had come up with this image right there. Very famous image in biology. It's called Photo 51. So Wilkins showed this to Watson, and Watson and Crick had enough of the pieces of the puzzle that when they saw this picture, they were able to figure out the rest, which is really cool. So by looking at this image, Watson was able to figure out that the DNA double helix had a uniform diameter of about 2 nanometers. A nanometer is 1/1000th of a meter, very small. So for comparison, a cell membrane is about 4 times thicker than that. And cell membranes are incredibly thin. So the diameter here suggests that the helix was made up of 2 separate strands, which, I mean, everybody knows that now. It was a double helix. So using this picture and what he already knew, or what they already knew, they were able to figure out that the sugar phosphate backbone must be on the outside of the double helix, not the inside. And they were able to tell that the single ring bases, the pyrimidines, always bonded with double ring bases, purines. Anyone guess how they were able to figure that out? It's the uniform diameter. So if it had been the other way around, they would have seen a much more variable diameter in the double helix, but they didn't. Because a pair of purines would be almost twice as wide as a pair of pyrimidines. Kind of one of the big dramas in the history of biology, too, was Rosalind Franklin never gave Wilkins permission to show them that photo. But they didn't really like each other, so he did it anyway. So a lot of people feel like she got hosed, and she probably kind of did. There was also a guy, another American, who was hot on the chase, and he didn't get to go to that conference because he was an outspoken anti-nuclear advocate back in the 50s. So the government suspended his passport as punishment. A lot of drama. He ended up getting a different Nobel Prize, actually 2 other Nobel Prizes. Anyway, so as it turns out, the matching of base pairs gets even more specific than just purines bonding with pyrimidines. I talked a little bit earlier about how the functional groups from each one will best pair with one specific other one. So you always get the A's bonding with the T's, and you always get the C's bonding with the G's. This is what complementary base pairing is.
So if there's this many requirements for what things get bonded to, how do we get such variety in the information?
So the answer is exceedingly long chains. So with 26 letters in the alphabet, we have who knows how many words, right? Now, we only have 4 letters to work with in DNA, but the words can also be tens of thousands of letters long. That's how you get huge variety. So complementary base pairing just means that each base has a complementary base that it always pairs with and will never pair with anything else. Does that make sense? That is one of the absolute most important properties of DNA. It becomes extremely important when we start talking about how it copies itself, a process called DNA replication. But moving on, talking about the structure again. So a good analogy of the double helix is just a twisted rope ladder. So you take a rope ladder, twist it around, that's basically what DNA molecule looks like. In this analogy, the rope would represent the sugars and phosphates that bond together to form the background, or the backbone. And the wooden rungs in the middle would represent bonded pairs of nitrogenous bases. I probably didn't need to show you guys this image, but just in case anyone was having a tough time conceptualizing the double helix, there you go.
Do we know the reason why it twists? Like for space?
I'm sure we do. I imagine it has something to do with compact, yeah. Mostly it would probably be just to save space. So DNA actually ends up getting supercoiled as well when it's not being actively used or copied. So it coils up and then it coils up again. Like you ever twist a piece of string so tight that it starts doing a double coil? That's what you see there. So here's a slightly more biologically accurate representation. The figure on the right shows the detail of hydrogen bonding between complementary pairs of nitrogenous bases. So if you look here, adenine and thymine form 2 hydrogen bonds. Cytosine and guanine form 3. That's what determines whether or not they pair with each other. Also, note that due to the fact that it's 3 hydrogen bonds instead of 2, it's a slightly stronger attraction. Anyone have any questions about that?
Those four nucleotide, believe they have to divide into 26 characters, 26 genetic characters.
No, that was just the number of letters in the English alphabet. I was making an analogy for trying to explain how we can have so many combinations with only 4 different.
So it's a 4, we can say 4 letters.
Yes, exactly. So all the words in DNA have to be spelled with only 4 letters. All right. So Watson and Crick published their findings in 1953. In 1962, they ended up finally getting the Nobel Prize because the wheels turned very slowly in this sort of thing. Maurice Wilkins also got one because he sort of undercut Rosalind Franklin and gave him that picture. Rosalind Franklin did not get one, and a lot of people mistakenly think that was due to sexism at the time, but it was actually due to the fact that she was already dead. She died from ovarian cancer at the age of only 37 because the early pioneers of X-ray crystallography did not realize how dangerous X-ray radiation was. So there was a lot of exposure to heavy radiation, so a lot of them died of cancer very young. And they never gave the Nobel Prize posthumously. So if she'd still been alive, I'm sure she would have. Now there's a key line in the paper that Watson and Crick published. Imagine me saying this with a really stupid English accent. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. This is talking about complementary base pairing. So by this point, they already knew that specific base pairs would only pair with specific other base pairs. And these guys were smart enough to think that this is probably important for DNA replication. And they were quite right. So in DNA replication, we have our DNA molecule. Our parent DNA molecule that needs to be copied. When does this happen? Yeah, the S subphase of interphase in the cell cycle, be it mitosis or meiosis. So if you understand the concept of complementary base pairing, the process of DNA replication starts to seem very simple and very predictable. If you know the order of nucleotides in one strand, you can very easily predict the order of nucleotides in the other strand, because you always know that A is going to be bonded with T, and C is going to be bonded to G and vice versa. So when that parental DNA molecule needs to get copied, what do you think happens? It unwinds, and then it separates, it unzips. There's a really bad pickup line that used to get tossed around in the bio department when I was in college. The enzyme that actually unzips the DNA molecule is called DNA helicase, and it's a really corny pickup line about something DNA helicase, I want to unzip the genes. Come on, fine. Anyway,
You're so happy.
in order to copy itself, that DNA molecule has to unwind and then it has to split. Once it splits, each strand becomes a template or a set of instructions for the assembly of the new strand that it will be paired with. Nucleotides are added from a supply that's just always floating around in the nucleus waiting to be used. And they line up one at a time in accordance with our complementary base pairing rules. So when you split this parental molecule, you have two single strands that can then basically just be instructions for which ones to stick here. That's how complementary base pairing helps to direct DNA replication. When you're finally finished with this process, you end up with two complete DNA molecules, which are called daughter strands of DNA, which, just like in the case of daughter cells, is not meant to imply gender in any way. Each of the two new copies will be genetically identical to the parental DNA molecule that existed before replication. Furthermore, each of the two copies will now have one strand of the parental molecule and one strand of new nucleotides. So we say the process is semi-conservative because when it's all finished, in the final product here, each molecule has conserved half of the original molecule. Does anyone have any questions on this? Yes.
So then back to like this question where how does variety go into play? Because like you said, the word length thing or whatever, like the length of chain, like you can't add length to the DNA thing because it all comes from one.
Right, but the length is already there. So when the chromosome is being copied, you already have this impossibly long polynucleotide.
Right.
So when you're copying them, nothing gets added to the length, but the length is already enormous.
Right, but it's the same length or whatever as the person that was copied what was changed.
So nothing gets changed here. This is just DNA being copied to pass it on to a new cell. So it's an already functional, already existing, already living cell that's splitting into two cells, and each of those two cells needs to have its own copy of the DNA. So this is meant to give an exact copy with no variation whatsoever. Does that answer your question?
I think I was on somewhere else because I remember you said like connectoration happens when chromosomes cross over, right?
Yeah. So if,
That's what I was on.
yeah, that happens after the copy. So after the chromosomes are copied during the S phase of meiosis, then you would have these completely finished copied daughter molecules of DNA, which could then meet each other through synapsis in metaphase 1 and switch tails. That actually? Okay. All right. Now, this seems like a really simple process, and conceptually it is. It's not too hard to understand what I've just explained, right? I hope. The basic concept of complementary base pairing and directing which nucleotides get added to the other nucleotides, right? That makes sense. I hope. As it turns out, though, the overall process of DNA replication is extremely complicated. It requires the coordination of more than a dozen enzymes and a whole raft of other proteins. The helix has to untwist and it has to unzip before it can be copied. That requires multiple enzymes. The new strands must be made simultaneously and also in opposite directions. I'll explain why that is in a moment. I'll probably use a video because it's going to be easy if you can watch it in motion. The process is also exceedingly accurate. So there's an error rate of about 1 in a billion in DNA replication. That is incredible accuracy. To make things even more complicated, DNA replication proceeds in 2 different directions, starting from many different sites simultaneously. So replication will begin at special sites that are at least understandably called origins of replication. And these are basically short stretches of DNA that have a specific sequence of nucleotides that allow a specific protein to attach and separate the strands. Then proceeds in 2 directions from those sites, creating bubble-like shapes. So up at the top here, we have our parental DNA molecule that's going to get copied. So at multiple different places, this protein will bind, unwind, unzip, and then DNA replication will continue in both directions from that site until they eventually meet each other, and we have our 2 daughter molecules of DNA. In eukaryotes, the DNA has a whole lot of different sites where replication occurs. Hundreds or maybe thousands of origins of replication. The point of this is just to reduce the amount of time this process takes. If it could only start at one end and go all the way through with one origin of replication, this would take forever because these polynucleotides are insanely long. But you start from 1,000 different sites, then those bubbles merge with each other pretty quickly, and the process is much more rapid. All right. Now, I mentioned a moment ago that the nucleotides have to be added to the new DNA chains in opposite directions. There's a reason for this. So, the DNA strands in the double helix run in opposite directions. Each one has a 3' end and a 5' end. In chemistry, whenever you see a number like that followed by an apostrophe, that's how it's read. 3', 5', etc. Now, what those numbers actually refer to is specific carbon atoms in the sugar molecule. So you have, where's that? Hopefully I still have my pen in here. Yes. All right. So if we were to look at one of these sugar molecules, remember we have that pentagonal shape, oxygen on the top corner, carbon, 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 carbon. So you start near the top and you name them counterclockwise. Every corner here represents a carbon. I'll write the C's there just so you know. That's how you draw it in organic chemistry, so it simplifies things. This is the 1' carbon, 2', 3', 4', and 5'. Always goes in clockwise order. So when I say the 3' carbon, I'm talking about this one. Or when I say the 5' carbon, I'm talking about that one. And as it turns out, molecules can only be added to that sugar phosphate backbone in one direction. They can only get added to the 3' end, never to the 5' end. And what that refers to is which carbon from that sugar molecule that's closest to the end. So in this case, you see the 3' carbon right there. And the 5' carbon is the one sticking out on that line right there. The opposite strand goes in the opposite direction. 5' end here, 3' end there. I know this is kind of confusing, so if anyone has any questions, please don't hesitate to ask. Don't worry about why things can only be added to the 3' end, just remember that they can only be added to that. And this is the reason why DNA replication has to continue in opposite directions. It can only grow the 5' to 3' direction. So you can probably guess, if you're picturing DNA replication, that this makes things more complicated. Because the replication fork only goes in one direction. So the 2 strands of the parental DNA molecule are getting separated in one direction, yet DNA synthesis has to occur in both directions. So here we see the action of yet another enzyme. As the strands get separated and get copied, one of them, you can see, is getting copied in one nice long continuous strand because that's the lucky one, and the replication fork is moving in the 5' to 3' direction for that strand. The other one, replication fork is going this way, but DNA synthesis can only occur this way. So what that results in is you have several discontinuous strands of DNA being made on that side. Those discontinuous strands are called Okazaki fragments, named for a married couple of Japanese scientists who discovered them. But you can't just have those discontinuous fragmented strands of DNA in a double helix, right? I mean, yeah, you can. So they have to get joined together. That job is done by another enzyme called DNA ligase. Now I know this probably seems really confusing. So I'm going to try to find a good YouTube video, because this is one thing where seeing an animation makes it a lot easier to conceptualize.
When DNA replicates, its strands are separated by the enzyme helicase.
Okay, that's insanely low resolution.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of the replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
You already did that. Okay. Why is autoplay always the default? Anyway, does that make it a little easier to understand? Do you have a question?
Yeah. So both sides have fragments?
So Okazaki fragments only get generated on the lagging strand, but the ligase is still used to bind the two new strands together. So when they said fragments in that case, they were just talking about two different strands. Yes.
Is there, can you remind me why the replication goes in opposite directions?
Because molecules can only be added to the 3' end chain. That just has to do with the properties of that sugar molecule and whether or not it can accept a new bond.
Okay. And the leading strand only needs one primer while the other one needs the primers and the glue.
Yeah. So DNA polymerase always binds to a primer that's already been laid down, and that's where replication starts. Then after that's done, the RNA primer gets removed and replaced with DNA and then it gets sewn together.
Basically, it's art.
Good question. Any other questions? Okay, let's get back to it. So hopefully this looks a little bit less confusing to you now. Makes more sense? If there's any more questions, keep firing. Yes?
Is there a strand that's always the leading strand, or does it change?
So the one that's going in the 5' to 3' direction, that's always going to be the leading strand. Okay. Anything else before we keep going? Okay. So, now what does the DNA get used for? Now that we've seen how it's copied, we've seen how it's put together, we know how it gets passed from one cell to the next in cell division. What does it actually get used for? What's the purpose? Exactly, coding for proteins. So when you talk about gene expression, to express a gene just means to make the protein that that gene codes for. So gene expression involves taking the DNA template, making an RNA template from that, and then making an amino acid from that. The process of making RNA from DNA is called transcription. the process of making proteins from RNA is called translation. So you can say the genetic information sort of flows in the direction of DNA to RNA to protein. Always goes that way.
Is transcription similar to?
Transcription is very similar. Yeah. Transcription is very similar to translation, and we're about to go into that in a little bit more detail. So this is the process of making that RNA template from the strand of DNA. This has to be done because the chromosomes do not leave the nucleus. They can't travel outside the nucleus to the site of protein synthesis. So instead, we make this RNA template, which can be sent out of the nucleus to the site of protein synthesis. And it follows the same basic rules of complementary base pairing as DNA. Only now, instead of making 2 strands, we're just making 1. Because remember that RNA is a single-stranded molecule. What's the other big difference? Yeah, we have uracil instead of thymine now. So wherever we have C, instead of a T bonding to it, it's going to be a U. When it moves from transcription to translation, after we already have that RNA template, that's when we start adding amino acids. So here we're building a polypeptide chain. A polypeptide chain is the basis for a protein. It's called polypeptides because the individual amino acids are joined to each other using peptide bonds. That's why it's not called polyamino acid. Also, because that sounds really funny. So as it turns out, 3 consecutive nucleotides in an RNA molecule form something called a codon. And a codon, that little sequence of 3 nucleotides, codes for a specific amino acid. So for every 3 nucleotides in that chain of RNA, we get 1 amino acid. The order of the codons, obviously, determines the order of the amino acids in the polypeptide chain, because they're added one at a time as that RNA template is read in a continuous line. Does this make sense to everybody? Okay. So we, quote unquote, cracked the genetic code in the 1960s. What that means is we know which codons translate into which amino acids. So we can then compile that information into sort of a genetic dictionary, which gets arranged into table form. And you can use that to tell exactly what the order of amino acids is going to be, given any sequence of RNA that I were to throw at it. As it turns out, most amino acids have 4 different codons associated with them. For instance, if we're talking about glycine in the bottom right here, a codon of GGU, GGC, GGA, or GGG will all code for glycine. Glycine is a specific amino acid. I'm not making you guys remember all the amino acids because this is a class for non-majors. But I had to do that in college. Thank you. So in eukaryotic cells, transcription obviously occurs in the nucleus because that's where the DNA is kept. If we're talking about prokaryotes, nucleolar. Using that DNA in the chromosomes as a template, the RNA nucleotides get linked together one at a time by the enzyme RNA polymerase. So it looks quite a bit like DNA replication. You still have the DNA template getting unwound and unzipped, and you still have an enzyme binding here and adding new nucleotides, The only difference is now you're only making a single strand, and the two unzipped strands will get rejoined. As that continues, you end up with a longer and longer chain of RNA. It starts out phase 1, called the initiation. Predictably, that's just when the RNA polymerase gets attached to the promoter, and that represents the beginning of RNA synthesis. The second phase, which is also pretty intuitively named, is called elongation. That's just adding more nucleotides to our growing RNA chain. As it grows, you can see in that image, it peels away from the DNA template, so that the two strands of the DNA molecule can be rejoined to each other. The third phase, called termination, you can probably guess what happens there. So that's when you have a specific sequence of DNA bases that just signals the RNA strand to detach. Or the RNA polymerase, sorry, to detach. At that point, the completed RNA strand then leaves the nucleus, travels out to the ribosomes, which is the site of protein synthesis, where it can then guide the protein synthesis happening in those ribosomes. So my favorite analogy to use is that the DNA is basically the master plans in the architect's office that never leave the architect's office, which is the nucleus. The RNA is the copied blueprints that get sent to the construction site. And unfortunately, there's 3 different types of RNA. This type is called messenger RNA, or mRNA, which is kind of intuitive because it's carrying the plant. It's the messenger of the plant. That's why they call it that. So the primer is not, don't worry about that for now. That won't be on the test. But I like curiosity. So now how do we know how the amino acids find their place along the RNA template during protein synthesis? This should have been done in progressive disclosure. I took way too much text after once. But we'll take it one piece at a time. So you have this mRNA molecule, this messenger RNA molecule, that's been copied in the nucleus and has now been sent to the ribosome and is then going to be used as the blueprint for the construction of the new protein. So how do the new amino acids find the right spot on the mRNA template? Turns out that has to do with complementary base pairing and another type of RNA, which is called transfer RNA or tRNA. So the amino acids themselves can't directly recognize the codons on the mRNA. That's what we have the tRNA for. So tRNA molecules will bind to a specific amino acid, and then they will sort of read the mRNA template based on the property of complementary base pair. So tRNA molecules have their own sequence of free nucleotides. That allows them to bind to the correct site on the mRNA and bring their associated amino acid in with it. Hopefully this is going to seem less confusing when I show you this picture. This is what the structure of tRNA looks like. So on one end, it has this specific amino acid attachment site. That will always bind to the appropriate amino acid with the aid of yet another specific enzyme that I'm not going to make you memorize. It also has something on the other end called an anticodon. So the anticodon is the opposite, or the complementary base pairs, for the codon on the mRNA. So that's how the tRNA molecule will recognize the right spot on the mRNA molecule to bring in its amino acid. That's how it gets added in the correct order. I feel like a YouTube animation might be useful here as well. Everyone agree? Okay. This one, right?
You write anything on your computer?
Wow, 1 minute long ad.
I write pretty much all day.
Ready to get one across in 5 seconds?
Steps of the translation process. Initiation. The small ribosomal subunit binds to the initiator tRNA carrying the initiator amino acid in the
This one has to be a little bit beyond the scope of this
This is
class.
complex, then it's
We'll find a simpler one. Maybe this one.
a transfer RNA molecule is composed of about 75 nucleotides. Each transfer RNA molecule recognizes only one amino acid, which becomes bonded to the top of the transfer RNA molecule. Located on the bottom of the transfer RNA molecule are 3 nitrogen bases called an anticodon that pair up with messenger RNA codons during translation. As translation begins, the first codon of the messenger RNA strand attaches to a ribosome. Then, transfer RNA molecules, each carrying a specific amino acid, approach the ribosome. When the transfer RNA anticodon pairs with the messenger RNA codon, the two molecules join. Often, the first codon on messenger RNA is AUG, which goes for the amino acid methionine. AUG signals the start of protein synthesis. The messenger RNA then slides along the ribosome to the next codon, where a new transfer RNA molecule carrying an amino acid will pair with the messenger RNA codon. The two adjoining amino acids then become joined by a peptide bond, and the first transfer RNA molecule is released by the messenger RNA. As the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon on the messenger RNA strand.
That make it easier to understand? You can watch this one too. About 15 seconds long. Yeah, that's probably useless. Well, anyway, did that last one help at all? Okay, good. Yeah, more enzymes. So this entire thing involves an incredibly complicated orchestra of dozens of different enzymes. So I will frequently put a true or false question on an exam asking whether or not DNA replication or translation or transcription is a simple process involving only one enzyme. You should all know by now that that is false. Okay, so a little bit more about ribosomes. Hopefully we'll have just enough time to finish this up. So ribosomes are the actual site of translation. Remember, translation just involves protein synthesis, and ribosomes are the site of protein synthesis. So their main job is to position the mRNA and tRNA close together to allow for the catalysis of polypeptides. No, wait. Never mind. It allows them to make polypeptides. Okay, it's the opposite of the right word. So a ribosome generally consists of 2 subunits, and it has yet a third type of RNA. I promise this is the last one I'll throw at you, called ribosomal RNA. So now if you ever have to give me an example of a type of RNA, you have 3 different ones to choose from. Messenger RNA, transfer RNA, and ribosomal RNA. So amino acids then get added one by one to the chain in a 3-step process, which we already saw in that video. Remember, the ribosome will recognize the codon in step 1. Then the tRNA brings in the correct amino acid, and a peptide bond forms between adjacent amino acids in the chain. And then something called translocation happens, which is where the ribosome just moves one step farther down on that RNA chain. So codon recognition. This is when the anticodon of that tRNA molecule pairs with the corresponding codon on the mRNA molecule. Hopefully, everybody understood all of those words I just said. If not, please ask me a question. Yes.
tRNA has the anticodon.
Yes. Yeah, so the codon, you remember what codon is, right? So no, codon is just a sequence of 3 nucleotides in that mRNA chain. The anticodon exists on the tRNA molecule, and it's called the anticodon because it's the opposite or the complementary pairs to go with that codon. That's how the tRNA molecule finds the right place in the chain. I wouldn't say it's necessarily misleading because it's the, yeah, I mean, can you think of a better word for it?
No, I couldn't, but it makes me think of, again,
Well, I think they call it anticodon because it goes opposite of the codon. They could have called it mirror codon or something, maybe, I don't know. Anyway, so the ribosome will then catalyze the formation of a new peptide bond, joining those 2 amino acids together. That's what happens during elongation. After that, anybody remember what the next step is? Translocation. So in translocation, that ribosome will just ratchet down one more codon on the mRNA template, grab another tRNA molecule, bringing another amino acid in line, which then has another peptide bond formation, then translocation happens again. And this just repeats over and over, all the way down the line, until the ribosome reaches that stop codon, which signals it to detach, and then you have your complete amino acid chain. Yes?
Is the protein functional, or does it have to be folded?
The protein will generally have to be folded. I don't know of any example where a protein doesn't get folded afterwards.
Is it another ribosome that does it?
It's not the ribosomes. A lot of the folding happens automatically due to the chemical properties of the individual amino acids. So we'll get, actually I don't know if we'll get to proteins this semester or not, but hopefully we will, and then I can explain a lot more detail about that. All right, anyone have any other questions?
Are the anticodons on the tRNA pre-printed or?
And they stay.
Okay, so the same enzyme is used for all the tRNA.
So, yeah, basically. So every tRNA molecule will always have that same 3 nucleotide anticodon.
Right, so how does the enzyme know which one you get?
So they're brought into the ribosome, and then it's just due to the properties of complementary base pairing and chemical attractions between those complementary nucleotides. So it'll just keep like bumping its head against the wall until it finds where it fits. Any other questions? Okay, that's as far as I wanted to get today. So the exam will cover the entirety of this chapter. And we've also got time to hand back the quizzes from last week. And we will also be having office hours starting as soon as I'm done passing out the quizzes if anyone has questions.